Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR PYQs of December 2023 Shift 2. I will be dealing with the topics from biochemistry, cell biology, genetics, applied biology methodology, DNA replication, protein synthesis, cell signaling. This is part C questions that I am discussing, and this is part 2 of my discussion. Then next one. Various type of processed eukaryotic endogenous mRNAs are mentioned below. Choose the option that identifies all the mRNA species that can be exported to the cytosol. So for an mRNA to be exported to the cytosol, it should be spliced, it should be polyadenylated, and it should have a cap. So unspliced and polyadenylated, no. Polyadenylated, okay. Capped, okay. Unspliced again. So it is also not possible to transport or export this mRNA. That is B mRNA. C mRNA, if you see polyadenylated, spliced, capped mRNA, that is correct. D is spliced, uncapped. So that is also not possible. So answer is option 3, C only. Then next one. The eukaryotic mRNA shown schematically in the diagram below has 5 ribosomes carrying out translation. Which one of the following statement about this polyribosome complex is true? So if you see the picture here, this is the 5' end and this is the 3' end. This is the mRNA, the blue one, it is the mRNA. These are the polysomes that are attached. If you see over the synthesis, you can see that this ribosome is nearest to the initiation codon. It is having a shorter polypeptide, whereas as you go from 5' to 3' end, the polypeptide chain length also increases. Okay. Now coming to the statements. The mRNA was being transcribed when the first ribosome started translation. Incorrect. The question is for eukaryotes, and but they have given the statement from prokaryotes because they have given transcription and translation is coupled, and that coupling is seen only in prokaryotes, not in eukaryotes. So first statement is not correct. Ribosome 5 is nearest to the initiation codon. Ribosome 5 is over here at the 3' end. At the 3' end, I told you it will have a longer polypeptide chain. Whereas at the 5' end, it is closer to the initiation codon and it shows short polypeptide chain. So ribosome 5 is nearest to the initiation codon is not correct. It is ribosome 1 which is nearest to the initiation codon. So 2 is also not correct. The polypeptide attached to the ribosome 4 is longer than that attached to ribosome 3. That is correct. If you see this picture, you can see as the ribosomes move from 5' to 3' and the polypeptide chain also increases in length. So 3 is correct. All the ribosomes have incorporated the carboxy terminal amino acid. That is also not correct. It is the N-terminal amino acid of the polypeptide that is incorporated, whereas C-terminal has not been released itself. The protein itself has not been released from the ribosome. So C-terminal end has not come. So the answer here is option 3. Then next one. Following statements are made about DNA base excision repair. Base excision repair process begins with DNA glycosylase, that is correct, which extrudes a base in a damaged base pair, then clips out the damaged base. So A is correct. First DNA glycosylase acts and it removes the modified base. In bacteria, DNA polymerase 3 fills in the missing nucleotide in It is DNA polymerase 1, not DNA polymerase 3. So B is not correct. Eukaryotic apurinic, apyrimidinic, apendonuclease performs proofreading activities. That is also correct. It removes the sugar phosphate bond or the phosphodiester bond, and it also has proofreading activities. Then APE1 possesses 5' to 3' exonuclease activity. AP1 is AP endonuclease. They have already given in option C as AP endonuclease, and here they have asked exonuclease. So it is not correct. It is AP endonuclease that is correct. So the answer here are A and C only. So that gives option 2 as the answer. So this is the base excision pathway. Here you have got a modified base. The modified base is removed by the DNA glycosylase. This will create an AP site. Then AP endonuclease will act and it will cut the phosphodiester bond. Then what happens is the DNA polymerase will add new nucleotides to the exposed 3' OH region, replacing a section of nucleotides on the damaged strand. Then the nick, this nick, it will be sealed by the DNA ligase and an original intact DNA sequence will be obtained. So that is what is happening in base excision repair. Then coming to next question. Two teams of researchers, 1 and 2, extracted chromatin from nuclei and examined them with an electron microscope. The team 1 found that isolated chromatin resembles beads on a string. In contrast, team 2 found that isolated chromatin appear as condensed fiber of 30 nanometer in diameter. So team 1 got bead on a string like appearance, whereas team 2 got condensed form. That means given below are few reasons for these different outcomes. Among the statements given above, the statements most appropriately defines the divergent outcome of team 1 and team 2. So team 1 isolated the chromatin with low ionic strength buffer and team 2 isolated chromatin with an ionic strength of approximately 0.15 molar KCl, physiological ionic strength. That is correct. That is why team 2 got condensed form and team 1 got beads on a string appearance. Then team 1 isolated chromatin with 0.5 molar KCl. No. So B is not correct. For team 2, the nuclear isolation method was not appropriate. No, that is also not correct. Team 1, chromatin got sheared during isolation. It didn't get sheared. So answer is A only. Answer is option 1. Then next one. As depicted in the figure below, the sequence of trympanosomal mitochondrial cytochrome oxidase subunit 2, COX2 mRNA does not match the sequence of COX2 gene. So here if you see the picture, this one, COX DNA and COX2 RNA, you can see so many U's coming in RNA, whereas the corresponding A's are not there in DNA. Now why is it? Because it is because of RNA editing taking place in trympanosomal mitochondrial cytochrome oxidase subunit 2 gene. Subunit 2 mRNA, not gene, mRNA. The mRNA contains 4 additional U's highlighted which are not represented by T's in the gene, and these 4 U's are presumably added to the RNA by editing. The following statements were made about RNA editing. Which one of the following option shows the combination of all correct statements? So RNA editing, it can be used to either add use to a mRNA or delete use from the mRNA. That I will show you in the next slide. So RNA editing can add or remove use from the target mRNA. That is correct. Editing occurs in the 5' 3' direction by successive action of one or more guide RNAs. Not 5' 3' direction, it is 3 to 5 direction. So B is not correct. A terminal U transferase, TUTase, facilitates addition of extra uridylates to the mRNA during editing. Yes, TUTase is involved in addition of uridylates to the mRNA during editing. The protein required for editing are encoded by mitochondrial DNA. No, they are encoded by the nucleus. So D is also not correct. So A and C are the answer. So the answer here is option 2. So this is what RNA editing is. So this is the guide RNA. This is pre-edited mRNA. This is endonuclease cleavage at the mismatch. This is guide RNA binding to the pre-edited mRNA. Then if you want uridine insertions, then you add TUTase and you will get uridine inserted over here. Then ligation and you get a uridine added mRNA. Then if you want uridine to be deleted, these are the use over here. They are deleted by exonuclease. Then ligation, this is guide ligation and you will get mRNA without use. So this happens in trypanosoma mitochondrial DNA. Then next one. Column X enlists some of the animal viruses and column Y enlists surface proteins that serve as their receptors. Which one of the following options represent the correct match between column X and column Y? So hepatitis A virus, it is alpha 2 macroglobulin A3. That gives you answer as option 1 itself. So if you know hepatitis A virus, you can get the correct answer. Then rotavirus. Rotavirus, if you see, it is acetylated sialic acid on glycoprotein. So if you know B also, then also you can get the correct option. Then poliovirus is immunoglobulin superfamily C1. So if you know C1 poliovirus, then also you can get the answer correct. Then rabies virus D2 acetylcholine receptor on neuron. So if you know rabies virus also, then also you can get the correct answer. So the answer here is option 1. Then next one. The table below summarizes various hormones in column X and induced cell responses mediated by them via cyclic AMP in column Y. Match all the correct combinations from the options given below. So this is the hormone epinephrine and major response is given in Y. So hormone epinephrine. Hormone epinephrine is glycogen breakdown A4. Okay, first option is not correct, whereas 2, 3, 4 we have to see. Then vasopressin is water reabsorption B3. That gives you 2 as the correct answer, whereas it is B5 in 3 and 4. Then glucagon is glycogen breakdown C4. That is correct. Luteinizing hormone progesterone secretion D2 and parathyroid hormone bone resorption. Okay, E5. So answer here is option 2. Then next one. G protein coupled receptors are the largest family of cell surface receptors. The GPCRs activate G proteins activate G protein. G proteins are usually composed of three subunits alpha, beta, gamma. The typical feature of these subunits and their receptor activation are given. Which of the following combination marks all correct statements? So coming to G protein coupled receptor, this is your membrane, this is G alpha, it is bound to GDP, so it is not bound to receptor. GPCR is not bound to receptor. Then G alpha and gamma subunits are membrane bound, whereas beta subunit it is bound to the gamma subunit. So once receptor bound, the G alpha will exchange GDP for GTP and it will become active and help in downstream signaling, whereas beta and gamma subunit they separate from G alpha. So this is what is taking place in GPCR. So G alpha is membrane bound and in an unstimulated state it binds to GDP. That is correct. Then G alpha is non-membrane bound. That is not correct. It is membrane bound. After binding to the ligand, GPCR acts like guanine nucleotide exchange factor and helps in G alpha activation. That is also correct. There is conversion of GDP bound to C alpha to GTP upon binding with ligand. Then RGS is regulator of G protein signaling and it acts as alpha subunit specific GTPase acting protein. That is, it converts GTP to GDP and it will inactivate the alpha subunit. So D is also correct. So the answer here is A, C and D. Answer 1. Then next one. Insulin receptor is activated on binding insulin molecules. This leads to the activation of the downstream PI3K pathway that triggers AKT to phosphorylate and inactivate a FOXO transcription factor. The lipid phosphatase P10 antagonizes the PI3K pathway. A reduction of function mutation in the insulin receptor dramatically increases lifespan of an organism. The following statements were made regarding the mutations and their outcomes. Which one of the following options represent the correct combination of statements? So this is what they have given. So here you have got the insulin receptor. When insulin binds to insulin receptor, PI3K is activated. It activates AKT and AKT phosphorylates and inactivate FOXO, a transcription factor, and the cell will die. But P10 it is having opposite action of AKT and the cell does not die. Sorry, not AKT's opposite action but PI3K's opposite action and the cell does not die. So the following statements were made regarding the mutations and their outcomes. A gain of function mutation in AKT makes the organism long-lived. How can that be possible? Gain of function mutation of AKT is over here and that doesn't make the organism long-lived because AKT will phosphorylate and inactivate FOXO and the cell dies. So first statement is not correct. A FOXO deletion mutation suppresses the long lifespan of the organism with a reduction of function mutation in the insulin receptor. A FOXO deletion mutation. So FOXO is mutated It suppresses the long lifespan of the organism. Of course, the cell will die with a reduction of function mutation in the insulin receptor. That is also correct. So B is correct. Then a P10 deletion mutation. So P10 is having a mutation, so cell will die because P10 is mutated, the cell will die. Okay. Suppresses the long lifespan of the organism. Yes. With the reduction of function mutation in the insulin receptor. That is also correct. So C is correct. Then, a loss of function mutation in the FOXO ortholog makes the worms long-lived. A loss of function mutation in FOXO. FOXO is mutated, so how can the worms be long-lived? No, the cell will die. So D is incorrect. So which are the statements that are correct? B is correct and C is also correct. B is correct, C is correct, A is not correct, and D is also not correct. So the answer here is B and C. Then next one. A cross was carried out between two strains of Neurospora carrying alleles capital A and small a. The cross led to the following octad patterns. These are the octad patterns. The numbers in the last row indicate the number of octads observed with the given pattern. So these are the numbers. This is the total. In what percentage of myocytes did segregation occur at anaphase 2? So these are the octads. So FDS ratio, that is first division segregation, means no crossing over happened between gene and its centromere. So segregation doesn't occur at anaphase 2. You get 4A and 4 small a. So if you see this one, capital A, capital A, capital A, small a, small a, small a, small a, small a, small a, capital A, capital A, capital A, that is 4A is to 4A ratio. So first two columns we need not have to take into consideration because they have asked for anaphase 2. Anaphase 2 segregation is second division segregation. In crossover between gene and its centromere, then capital A and small a allele do not get separated until the second meiotic division. This is called second division segregation, and you get these ratios: 2 capital A, 2 small a, 2 capital A, 2 small a. If you see this one, 2 capital A, 2 small a, 2 capital A, 2 small a. So this is second division segregation, that is 14. Then 2 small a, this one, 2 small a, 2 capital A, 2 small a, 2 capital A. Again second division segregation. Then if you see this one, 2 capital A, 4 small a, 2 capital A. Again second division ratio, that is 16. Then 2 small a, 4 capital A and 2 small a, 18. Again second division segregation. So now add up all the SDS values. So 14 plus 12 plus 16 plus 18 divided by total, they have given 400 into 100 will give you 15 as the answer.